Hello, 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 everyone. This is Dr. Garrett Smith here with another one of the big episodes of the Love Your Liver live stream. Today, we are going to be covering heart disease, which includes, strokes, heart attacks, anything else, cardiovascular disease related. So I'm trying to keep it mainly to, I was searching for cardiovascular disease. So it's going to include all of it. I'm not doing anything specific because, well, just like chronic disease, it's connected via these same mechanisms that we're going to go over today. So is cardiovascular disease. So if we're improving one aspect of cardiovascular disease with what we do, we're probably improving all of it. So let me get my stuff pulled up and ready. So I have it all ready here. Okay. So for those of you who don't know, I am Dr. Garrett Smith, known as the nutrition detective around these here YouTube parts. I kind of missed that part today. And what we are going to go over today is how the toxic bile paradigm and toxicities, various toxicities, and nutrient deficiencies, particularly mineral deficiencies and niacin deficiency, nicotinic acid deficiency, correlate with heart disease. And therefore, since these are things that we generally need to live, we can assume that they are what we call root causes of cardiovascular disease. Like nobody has a statin deficiency. Nobody has a nattokinase deficiency. Nobody has a vitamin A deficiency. We've been over that before, but we're going to go over, oh my goodness. After Mike Roberto on Twitter, I'll go over his tweet, but he went over this RBP4 study about heart attacks that was brutal. And then I went into the rabbit hole of my phone and searching for studies. And then I came up with about 5 or 6 more. It's brutal. Vitamin A is absolutely brutal on your heart and your cardiovascular system. So we're going to go over all that. Let me get into it now. So yes, here we go. Okay, one of the very first things, for those of you who are new to this channel, toxic bile paradigm, which used to be called toxic bile theory. If you want to get a basis on that, you're going to want to look at episodes 53 or 71 or both. You can start binge watching the channel if you'd like. Many people do, and we appreciate that. Oh, for those of you who don't know, we passed 10,000 subscribers this last week and a half, and we're already at 10,100. So we're gaining more subscribers now. A faster rate of gaining subscribers than we've ever had before. So I want to thank all of you for sharing the word. And yeah, we kind of passed that little milestone. So anyway, back to toxic bile paradigm. One of the ideas here is that, the main idea is that as your liver gets damaged and your detox gets slower and you get more toxic and you have more nutrient deficiencies, you start leaking bile into your bloodstream. Your bile is by definition the most toxic fluid in your entire body. That's how your liver gets rid of its waste, its toxicity. It doesn't just poof disappear. So, being that bile contains the most, anytime you think of detoxing, you have to think of bile. Bile is how your liver gets rid of the toxicity, okay? And if you leak it into your blood, you are self-poisoning. So we're going to see, is bile connected to cardiovascular disease? This is always the first thing we go over. Now, again, watch those two overviews of toxic bile theory, now called toxic bile paradigm, to get an idea of how this all works. I'm working on the book on toxic bile theory, a short book, so people can read it, so you could share it with your friends. It's not done yet, but we're working on it. Okay. So this paper, and these papers, Joe put up the link to a Google Doc that has all of these references in it. So I'm not going to put up each reference as we go. All the references are in that Google Doc. It's probably 4 pages of straight references. Okay. So here we have overview of bile acid signaling in the cardiovascular system. Let's go over what we know about bile acids in the cardiovascular system. In addition, increased bile acids, so they mean in the blood, or the serum, they'll call it, can impair cardiovascular function in liver cirrhosis. What is liver cirrhosis? Liver cirrhosis is scarring or fibrosis of the liver. The liver is actually kind of turning into basically the equivalent of a raisin. And the bile that it makes is basically leaking straight out into the bloodstream. So liver cirrhosis is the worst example of toxic bile leakage. And we have helped someone completely recover from liver cirrhosis based on testing and imaging. So are we fixing this problem? Yes, we are. So what do we see here? Increased bile acids in the blood can impair cardiovascular function in liver cirrhosis. Approximately 50% of patients with cirrhosis develop cirrhotic cardiomyopathy. That is a disease of the heart. So heart muscle disease. 50% of patients with cirrhosis leaking toxic bile into their blood develop the type of heart muscle disease associated with cirrhosis. Exposure to high concentrations of hydrophobic. This means scared of water. They don't like water. So they like fat. Bile acids, so these are lipophilic or fat-loving bile acids, has been shown to be related to adverse effects, bad effects, with respect to vascular tension. This is blood pressure. Endothelial function. This is the recovery or the healing ability or the function of like the inside of your blood vessels. Okay? Arrhythmias, irregular heartbeats. Coronary, this is related to the heart. Atherosclerotic, this is the plaques in the blood vessel and inflammation. Heart disease, so coronary atherosclerotic heart disease and heart failure. High concentrations of bile acids in the blood has been associated with all of these things. The bile acids in the serum bile acid pool, so the bile acids floating around in your blood, have relevance, have relevance through their hydrophobicity. So they don't like water, they like fat. And the lipophilic bile acids are more harmful to the heart. So let me go over this, folks. Lipophilic means fat-loving. So probably the bile acids that are fat-loving are more associated with, let's say, when you eat lots of fat, a mistake that people do, thinking that if they eat more fat, they're going to dump more bile. No, you're going to feed the bacteria that make more fat-loving bile acids in your intestines. We're going to go over that in the biome section, okay? So eating more fat is not how you fix your toxic bile problem. It is absolutely the opposite of what you do. You reduce your added fats to the minimal amount you need. You do not eat a fatty diet. This is why fatty diets are associated with ruining gallbladders and ruining livers. Don't do that. So let's keep going. I found this paper. This is not necessarily directly linked to heart disease. I mean, you'll understand it when you read it, but bile acid induced cell injury and protection. Ooh, let's go back one. Okay. Just listen to how bad these bile acids are. So let me go give you a quick overview. Where is bile supposed to go? Bile is made by your liver cells. That is supposed to go into the teeny bile ducts inside your liver, which then goes into your big common bile duct, which then is supposed to mainly it goes into the gallbladder or it goes into your small intestine. And the gallbladder, when it dumps, it squeezes, it goes into the small intestine, okay? That is supposed to go down the small intestine, combine with food and fiber and any other bile acid binders that you're taking, and you're trying to get rid of it the most, you're trying to poop it out. If you're not getting rid of it, or it's getting very, very toxic, you are reabsorbing a lot of it, or you're leaking it into your system. And when you reabsorb a lot of it in the intestines, it goes right back to your liver to just do the loop again. So think about it. The more it does this loop, it's called the enterohepatic circulation. The more it does the loop, the more concentrated in toxicity it becomes. It may be staying in that loop. That's kind of a closed loop. But over time, it will cause problems, and you're just concentrating more toxicity in your liver and in that loop. So let's go over what bile acids in the blood do. Several studies have characterized the cellular and molecular mechanisms of hepatocyte. This is liver cell injury caused by the retention or holding on to of hydrophobic, again, lipophilic. Wait, what do we have a problem with these days? Fatty liver. Okay, we'll hit that in a second. Water-fearing, fat-loving bile acids in cholestatic diseases. What is cholestasis? Cholestasis is the actual leaking of toxic bile into the bloodstream or anywhere else that it's not supposed to be. Up into your stomach, that's the cause of acid, what we call GERD, gastroesophageal reflux disease. Did you notice how they changed it? It used to be acid reflux. But then they changed it to gastroesophageal reflux disease, and they took the acid out of it. That's because it's a bile reflux disease, and you should watch my whole episode on that. Okay? I want you to go over, because we went over retention or accumulation or storage of bile acids in cancerous tumors was it last week, I believe? Yeah. So let me just make sure. Liver cells, the same liver cells that make bile, right? Shouldn't they be impervious to bile? Shouldn't they be not injured by bile? Oh, wait, bile actually injures the liver cells that make it. Okay. Caused by the retention or storage or accumulation or holding on to of fat-soluble bile acids. Well, wait, if they're fat-loving bile acids, if they're fat-loving bile acids, what type of tissue would the liver store them in? Fatty tissue. What do we have a problem with again? Fatty liver. What is vitamin A as a toxin? Is it fat-soluble or water-soluble? Oh, it's fat-soluble. If you had too much vitamin A in your liver, what would your liver store it in? Fat. What color is bile? What color is vitamin A? They both tend to be yellow. If you go and look up pictures of fatty liver on the internet, what color is fatty liver, folks? It used to be maroon, like a nice maroon color, and then in fatty liver, it turns yellow. Okay? Don't have to be a genius to figure out these associations of color and fat and what's happening. Okay? Bile acids may disrupt cell membranes. That's bad. That's like leaky cells, through their detergent action on lipid components, fatty components, and can promote the generation of reactive oxygen species, those are bad, that in turn oxidatively modify lipids. We'll go over malondialdehyde, proteins, and nucleic acids, and eventually cause hepatocyte liver cell necrosis, death, and apoptosis, programmed cell death. Just death and wasting away, or programmed cell death. I want you to also pay attention to detergent action. When people say bile acids are emulsifiers, no, a detergent is it's more of a chemical action, and it's more than emulsifying. They're saying here that bile acids disrupt cell membranes through their detergent action, not through their emulsifying action, okay? Emulsifying just makes water and fat able to mix. Detergent action, they don't call your laundry detergent laundry emulsifier. They call it detergent because it's a chemical action, and it's way more than emulsification. Kills liver cells. Okay. Several pathways are involved in triggering hepatocyte apoptosis, programmed cell death of liver cells. Toxic bile acids can activate liver cell death receptors directly. That's weird. That seems like a poor design. And induce oxidative damage, thereby causing mitochondrial dysfunction. Go ask all the mitochondria nerds how much they know about bile acids ruining your liver cells and killing your liver cells. I'm so tired of the mitochondria nerds who just look at cell studies and think that things that help the mitochondria in a cell study are going to work the same in humans, because the duration paradox says it's not true. And induce endoplasmic reticulum stress. When these compounds are taken up and accumulate inside biliary cells, when these toxic bile acids are taken up and accumulate inside the bile duct cells, they can also cause apoptosis. So one of the places you leak bile into your system is through the bile ducts. How could we create holes in the bile ducts and cause leaky bile ducts? Oh, I don't know, if the bile acids were accumulating inside the bile duct cells and causing programmed cell death. How bad are these toxic bile acids when they destroy the very cells that make them and the very highway that they're supposed to that is designed to have them travel down? Regarding extrahepatic tissues, this is everything except the liver. The accumulation of bile acids, wait, there it is again, the accumulation of bile acids in the systemic circulation, so they're going higher and higher in the bloodstream, may contribute to endothelial injury in the kidney and lungs. In GI cells, gastrointestinal cells, your gut, bile acids may behave as cancer promoters through an indirect mechanism involving oxidative stress and DNA damage, as well as acting as selection agents for apoptosis-resistant cells. The accumulation, there it is again, of bile acids may also have deleterious effects on placental and fetal cells. You know that 80% of kids these days are being born with jaundice in the Western world? What is jaundice? It is leaking bilirubin, also known as, the way you can pronounce it is bilirubin, into the blood, and it's accumulating in the skin. So, if you were born with jaundice, or if your children were born with jaundice, I have some news for you. That person's liver was damaged when they were born. Like, they were born with a damaged liver and bile ducts and all that stuff already. They are leaking bile into their bloodstream. Yeah, you could do UV light to get rid of the coloration. Does that mean you fixed the liver? No, it doesn't mean anything about you fixing the liver. So if you've had health problems your entire life and you were born with jaundice, there's a reason why you've had problems your entire life. You've been leaking bile your entire life. Okay, so I just wanted to go over this one. Boy, that's pretty nasty stuff, isn't it? Moving on. Overview of bile acid signaling in the cardiovascular system. Some of you know where I'm going with this already. Whenever researchers tend to use the word, okay, don't send super chats today. We're saving all super chats for next week. So super chats are for next week. I'm not going to have enough time. I got too much stuff to go through today. So overview of bile acid signaling. Whenever you see the word signaling, they're saying it's toxic, and they're going to try to convince you that it's good, even though they're going to show you all these ways that it's bad. Signaling is scientific gaslighting, where they have to say that it's good, but they're also going to say it's bad at the same time. So whenever you see this word, your alert should come up as, this is probably toxic. Now, I saw in another paper, they said zinc was a signaling molecule. And I'm like, zinc is essential. Zinc is not a toxin. You could get too much zinc, don't get me wrong. But if you don't have enough zinc, you're going to accumulate all sorts of other toxicity like cadmium and copper and all that stuff. So you kind of pick which one you need. And since zinc is involved in, what is it? Like, the last count was 400 enzyme pathways in the human body. It's probably important. So let's just review this again. In addition, increased bile acids can impair cardiovascular function, liver cirrhosis. Oh, wait, we already went over this one. Sorry. Oh, this is the same one. Okay, sorry. We're on the next one. Oop, I closed my share. Let me go do that again. I'm catching it quick, Joe. Let me do this. That's not the right one, Joe. Yeah, we're still only on the first window. So yeah, I'm not going to have time for any questions today. Okay, the role of bile acids in cardiovascular disease, from mechanisms to clinical implications. Studies have shown that cardiac dysfunction is associated with abnormal bile acid metabolic pathways. It's not that they're abnormal. It's that the things people are doing are making more toxic bile acids than less. Whenever you see people out there who don't understand what they're talking about, trying to talk about vitamin A pathways need to be fixed. No. That would be like saying your lead metabolism pathways need to be fixed. Your cadmium metabolism pathways need to be fixed. No, it's a toxin. They're doing apologetics. They're just trying to justify the fact that they still want to say that vitamin A is necessary, and they're desperate, and they've run into desperation. They're trying to explain that it's still necessary while understanding that it's toxic. So avoid these people. Anybody who talks about broken vitamin A metabolism pathways and you need to fix those, ignore them. Okay? As ligands, that means they bind to several nuclear receptors and membrane receptors. Okay, folks, just because something binds to something doesn't mean you need it. Okay? That doesn't imply that you need it. Bile acids systematically regulate the homeostasis. What's another word that we figured out they use to hide toxicity? Homeostasis. This paper has all the magic words. They have abnormal pathways, they have homeostasis, and they have signaling. Where was the signaling? There, wait. Anyway, I saw it before. You guys saw it too. Okay. They systematically regulate the homeostasis of metabolism and participate in cardiovascular diseases. They regulate the homeostasis of metabolism and participate. I'm pretty sure they mean cause cardiovascular diseases, such as myocardial infarction, heart disease, diabetic cardiomyopathy, that's the heart disease associated with diabetes, atherosclerosis, that's the good old plaques and heart disease, arrhythmia, heart rhythm problems, and heart failure. That's pretty bad when you get there. However, the molecular mechanism by which bile acids trigger cardiovascular diseases remains controversial. Do you really need to know what the mechanisms are? After they just told you, they're trying to do apologetics and say that it's good, but it's bad. You must watch out for these researchers who are trying to protect their jobs because they can't say that things are bad. Okay, so what happens when we start binding up bile? What an idea. We would never do such a thing. Effect of bile acid sequestrants on the risk of cardiovascular events, a Mendelian randomization analysis. So first, I want to say cholestyramine is the pharmaceutical bile acid sequestrant. We don't use it. I don't recommend using it. If you use it, like this would be like the people who think eating a ton of fat is going to help their bile problem. No, it's not. It's going to make it worse. Then the people who think cholestyramine, because they talk about it as a bile acid sequestrant in the research, is going to be good for them. No, it's a pharmaceutical. There's go look up side effects of cholestyramine. Go. I had one woman who came to me and she was sort of addicted to cholestyramine. And what I mean by that is she knew she felt worse on it, but she felt even worse taking it. So she was stuck and she didn't know what to do. And she was also stuck because she could only take a certain amount of charcoal before she'd start getting other problems. But she knew the charcoal would help, but it also caused other issues. She was stuck. Don't mess with pharmaceuticals. I don't care if they say it grabs onto bile. It causes other problems. So, yeah, that's what BAS means here. Bile acid sequestrants. So what did cholestyramine improve? It was associated with a reduction in LDL cholesterol. That's the bad cholesterol, right? Your body uses LDL cholesterol to bind to toxins, if you didn't know that. And a trend towards reduced risk of coronary artery disease. I believe that's CAD, right? That's what they're saying. Coronary artery disease, yes, right there. Oh, well. We'll just highlight it, and then we'll do that. There we go. Whereas colesevelam, I believe that's another bile acid sequestrant, was associated with a reduction in LDL cholesterol as well, based on findings that RS4299376, what is that? Must be I don't know. I'm not worried about it. It was associated with a 2.75 point decrease in LDL and a 5% decrease in risk of coronary artery disease outcomes. We estimated that cholestyramine was associated with an odds ratio for CAD, so it reduced the risk from 1 down to 0.63, and colesevelam with an odds ratio of 0.64. So that reduced the risk as well. Okay. Is it that the bile acid sequestrant is binding to the cholesterol, or is it that it's binding to the bile? It's that it's binding to the bile. So what we just saw was bile in the blood is associated with cardiovascular disease. This is how we can demonstrate these things when they don't have a direct study. We show that they know all sorts of bile in the blood causes heart disease. Okay, what happens when we reduce the bile in the blood? Does heart disease go down? Yes. Okay, we can be pretty sure of that. Okay, so what about something we use regularly? What about activated charcoal or activated charcoal, or what they call in the research activated carbon? What does it do? Well, it adsorbs bile salts as well. I'm not going to read this whole one. The results indicate that activated charcoal has a high capacity for bile salts, completely removing them from solutions of up to 5 millimoles and at a rate consistent with physiological activity. Finally, down here, based on these data, the adsorption of bile salts by activated charcoal appears to be a likely mechanism for activated charcoal-induced reduction of serum lipids. So it is the bile acid or bile adsorption that causes the reduction in serum cholesterol, okay, and triglycerides and all that stuff. So let's keep going. Effective activated charcoal on hypercholesterolemia. Has your doctor ever told your parents or you to go take charcoal for cholesterol? Probably not. Wow, this study was back in 1986. Does pharma make any money on charcoal? No. So I want you to pay attention to this, because there's really the only main side effect that you can get from charcoal, which is known, is constipation. Okay, so if you get constipation, you either have to take less charcoal or you need to take something to help you poop with it, like magnesium by mouth. Okay? Pay attention here. 7 patients with hypercholesterolemia were treated for 4 weeks with activated charcoal at a dose of 8 grams 3 times a day. Now, that's probably like 2 teaspoons 3 times a day. That's a fair bit of charcoal. That's quite a bit. That would stop up a lot of people. But I want you to pay attention here compared to statins. Plasma total cholesterol and LDL cholesterol decreased by 25% and 41%, respectively, whereas HDL cholesterol increased by 8%. Side effects were negligible. Why wasn't everybody using charcoal to lower their cholesterol? Now, for those who, so we're talking about, we just saw in that previous study, when they adsorb, when they suck up bile out of your gut and you poop it out, your cholesterol comes down. Cholesterol is made by your body as a protective lipid, lipoprotein, lipid protein. So one more time, let's go over this. Activated charcoal in the treatment of hypercholesterolemia, dose-response relationships. What that means is if they take more charcoal, they get a bigger response. And comparison with cholestyramine. So those of you who are thinking of doing cholestyramine, well, go look up the side effects. And then let's look at this paper in terms of how well charcoal works compared to cholestyramine. So in a crossover study, they had charcoal and finally bran. Oh, fiber. What an idea. Each phase lasting 3 weeks. So we saw total and LDL cholesterol went down 29% and 41% at the maximum in 3 weeks. And the ratio of LDL to LDL, so your good to bad ratio, was increased, which is good. 121% in someone by charcoal in a dose-dependent manner. Okay, 10 further patients with really bad cholesterol did it. The concentrations of total and LDL cholesterol were reduced by charcoal 23 and 29%, cholestyramine 31% and 39%, and their combination, so they mixed them, 30 and 38%. The ratio of good to bad cholesterol was increased from 0.13 to 0.23 by charcoal and to 0.29 by cholestyramine and to 0.25 by their combination. Serum triglycerides were increased by cholestyramine, but not by charcoal. I don't think you want to increase your serum triglycerides. Again, an L for cholestyramine compared to charcoal. Other parameters, including the serum concentrations of vitamin A, E, and D3, remained unaffected. The changes in lipids only partly subsided during the 3-week bran phase. So they maintained some of their improvements. In general, the acceptability by the patients and the efficacy of activated charcoal, cholestyramine, and their combination was about equal. The acceptability by the patients and the efficacy was about equal, but there were individual preferences for particular treatments. You go and you mess around with cholestyramine, folks. Don't blame me. Don't blame me. You can do charcoal and it's just as effective, and there's not pages of side effects with charcoal. And yes, you may need a little help pooping. Yeah. So now we're into the biome. So an important thing that you need to understand. Oh, let me share this tab. Sorry, Joe. Important thing you need to understand about bile is bile goes into it's supposed to go into your intestines. Now, what your bile is made of can both eat holes in your intestines or not. And what your bile is made of will feed your gut biome for better or worse. So if you say, my gut biome is all messed up and dysbiosis and whatever, okay, you are feeding that gut biome with your bile multiple times a day. Every time you eat, probably once or twice at night, like you are feeding your gut, you are making your gut biome into what it is. And I've gone over studies where they say that bile regulates the gut biome. Bile is the cause of your leaky gut. Bile is the cause of your messed up gut biome. The reason why people aren't fixing these things regularly is because they don't talk about these things, because they don't know about them, or they're denying it. So here's the paper, Nutrition and Gastrointestinal Microbiota. That's the microbiome. Microbial-derived secondary bile acids and cardiovascular disease. Secondary bile acids, folks, are generally regarded as more toxic, much more toxic than primary bile acids. Primary bile acids are made by the liver. Secondary bile acids are made by your gut bacteria. Okay? Secondary bile acids are considered worse. And then there's even tertiary or 3 steps down bile acids. Okay. Let's go over what they do. Bile acids exert a much broader range of biological functions than initially recognized. Well, we're recognizing them, that's for sure, including regulation of cardiovascular function. That means they make it better or worse. Through direct and indirect mechanisms, there is a bidirectional relationship between gut microbiota modulation or changing of bile acid signaling properties and their effects on gut microbiota composition. Okay, you understand? So it's like, okay, so get this. When they say bidirectional, okay, so the bile, what's in the bile will change your gut biome. Okay, and your gut biome changes the bile. Remember how I told you the enterohepatic circulation? So you change that bile, you make it more toxic generally in your gut. And it goes right back up through the enterohepatic circulation to your liver. So your liver sends toxic bile to your gut biome. Your gut biome can then make it more toxic, and then it goes back to the liver. This is a vicious cycle if you get into it. This is why the most important thing you can do to regain your health is to avoid taking in the toxins in the first place. We always prioritize stopping intoxing or reducing intoxing over pushing detox. Toxins you don't take in don't have to be processed. Evidence, primarily from rodent models and limited human trials, suggests that dietary modulation, changing your diet, which changes your gut biome, significantly impacts bile acid metabolism and subsequently host physiological responses. Available evidence suggests that the link between diet, which is what we change, right, gut microbiota, and cardiovascular risk is potentially mediated or controlled via bile acid effects on diverse metabolic pathways. Gosh, this sounds like the exact reasons why we help so many people with so many different conditions. Because we change their diet, which changes their gut microbiota, which changes their bile, and then we see less risk, or we see diseases just go away. Okay? Let's keep going. Gut microbiota derives secondary bile acids receptor polymorphism. So this would be genetic differences in your bile acid receptors and risk of cardiovascular disease in individuals with newly diagnosed type 2 diabetes, a cohort study. Secondary bile acids, the products of bacterial metabolism, like I said, are ligands, or that means they can bind of the FXR receptor and have been implicated. I love how they say this. You got to see how they're gaslighting you. They've been implicated in cardiovascular health. Wait, couldn't they have said they've been implicated in cardiovascular disease and it means the same thing? You could say something's implicated in disease, or you could say something's been implicated in health, and one of them is going to look much more positive and possibly mislead people, and one of them is going to look much more negative. Diet can modulate gut microbiota composition and bile acid metabolism. Let me just see if there's anything else. Okay. A higher proportion of unconjugated secondary bile acids. So these are bile acids that don't have anything bound to them, especially deoxycholic acid, is linearly associated with a higher risk of cardiovascular disease among people with newly diagnosed type 2 diabetes. Our findings support the potential role of a gut microbiota-derived secondary bile acids in cardiovascular health in individuals with type 2 diabetes. A higher proportion of unconjugated secondary bile acids means, and then when they say linearly, so they're just saying the more secondary bile acids, especially DCA, the higher the risk of heart disease. They're saying it's linearly associated. They move up directly together. They're like locked together. So, okay, so whenever you see medicine talking about therapeutic targets, you got to watch out. Gut microbiome derives secondary bile acids, therapeutic targets for reducing cardiovascular disease and type 2 diabetes. Well, based on the study we just went over, if they say it's a therapeutic target, they're saying secondary bile acids. They just said that they went up linearly together. So what do you think their therapeutic target is? To reduce it? Probably. We didn't have there was no abstract, but I just wanted to go over this because they just basically told you what they're going to try to do. And let me get back up here. This one, I'm going to have to do my little searches. Role of the gut microbiome in the development of atherosclerotic cardiovascular disease. Okay. Oh, yeah. I put this here because I wanted the people who think that doing more fat in their diet will somehow help their bile problem. Secondary bile acids, which are made by your gut, permit absorption of lipid nutrients. So, they help you absorb fat. Okay. And fat-soluble vitamins. Well, what do we know here about most what they call fat-soluble vitamins from food or supplements? They're toxic. So, secondary bile acids, which are made by gut bacteria, which these gut bacteria love fat, and they make lipophilic or fat-loving bile acids that you reabsorb. So the thing to remember is you're going to reabsorb those fat-soluble bile acids, those fat-loving bile acids, with the fat and the fat-soluble vitamins, also known as fat-storable toxins. So the more fat you eat, the more fat you're absorbing, the more fat-soluble toxins you're absorbing, the more bile you're reabsorbing. That's what all this means, okay? So no, extra fat is not the way out of this. I don't know how many ways I can say this. Okay, so let's keep going. Oh, vitamin A. I'm not going to spend too much time on this one, but this is going to be brutal, folks. Those of you out there who are pushing vitamin A and watching this, and you're trying to tell people that they need vitamin A for their heart, this is going to be brutal. Okay, I've been over this paper before. Increased dietary intake of vitamin A promotes aortic valve calcification in vivo, so in actual live animals. The more vitamin A they take in, the more their heart valves calcify. Dang. Calcific or calcified aortic valve disease is a major public health problem with no effective treatment available other than surgery. Well, magnesium helps. We'll go over that. We previously showed, so they already showed that mature heart valves calcify in response to retinoic acid treatment. Remember, folks, retinoic acid, I've shown research before that if you eat beta carotene from fruits and vegetables, it increases your retinoic acid levels. If you take in vitamin A from foods, supplements, liver, cod liver oil, whatever, any retinol, animal vitamin A, that increases your retinoic acid levels. We can show that in people eating food-based vitamin A, that they have Accutane, 13-cis-retinoic acid in their urine, in their blood. We can also show that when people are eating vitamin A from food, they have all-trans retinoic acid, which is also known as retin A, in their urine, in their blood. So if you are eating vitamin A on purpose or trying to eat a lot of it, you are increasing your retinoic acid. Same compound. You guys out there who say that natural retinoic acid is different than synthetic retinoic acid, first of all, the researchers who do these studies can't tell the difference. When they do all their fancy analysis, they cannot tell the difference. There is no difference. The burden of proof is on you guys to show how they're different. Because when they find retinoic acid in the pee of people who aren't taking Accutane or retin A, it's the same stuff. Calcifying heart disease. Okay, with retinoic acid. Well, what'd they give these mice? Retinol palmitate. Okay. Retinol palmitate is retinol bound to a palmitic acid. Retinol is animal vitamin A. Retinol palmitate, I need you guys to understand this so you can relay this to the people who deny this. The main form of vitamin A that is in animals, in milk, in cod liver oil, in the liver, is retinol palmitate. The main form of vitamin A that is put into milk, like fortified milk, or is in most supplements, is retinol palmitate. It's the same stuff. Chemically, it's the same stuff. Those who want to say it's different, the burden of proof is on them to show that it's different. And if they look at my natural versus synthetic vitamin A thread on Twitter, they're going to find out, and I think I have a video on the YouTube too, they're going to find out it's exactly the same stuff. And it does the same thing at the same doses. So what happens here? We show that mice fed excess vitamin A develop aortic valve stenosis. This is calcifying, and stenosis is especially when a tube, like a valve, let's say a valve that opens up, or your blood vessels, or the holes in your spine where you're, in your vertebra that your spinal cord goes through. Stenosis means that the hole is shrinking, like pinched nerves or like blood vessel disease, atherosclerosis, where it's the tube that stuff can go through is shrinking. That's what stenosis means. So it could be in a blood vessel. It could be in what's called a foramen of your spinal cord. It could be but anywhere in the body where a tube, where a hole that something passes through is shrinking. And calcification does this the most. Okay. And leaflet calcification, those are the, when you look at a heart valve, right, there's like 4 flaps. Those are the leaflets. So that's actually the valve. So they're saying the valve itself is shrinking, and leaflets, the parts of the valve that flap open and closed, are calcifying, associated with an increased expression of osteogenic or bone-building genes. Your heart's not supposed to build bone, folks. And decreased expression of cartilaginous markers. Wait, it should be a lot more like cartilage in there than bone. Using a pharmacological approach, I don't know what that means, we show that our retinoic acid-mediated repression and calcification is regulated by classical RA signaling and requires both retinoic acid and retinoid X receptors. Let's cover this again. What do we say signaling was? A sign of them trying to hide toxicity. Retinoic acid mediated or retinoic acid causing calcification is regulated by retinoic acid toxicity. And it requires retinoic acid and retinoid X receptors. So the receptors themselves are part of the calcification process. It sounds like bad design. And I joke when I say bad design. It's a toxin coming in, and toxins can bind to receptors. Our studies demonstrate that excess vitamin A dietary intake promotes heart valve calcification in actual live animals, therefore suggesting that hypervitaminosis A, or vitamin A toxicity, could serve as a new risk factor of calcific aortic valve disease in the human population. I think this one is pretty quick. The role of beta carotene and vitamin A in atherogenesis. Evidences from preclinical and clinical studies. Well, let's just look at that title, right? The role of it in causing atherogenesis, disease of your blood vessels. ASCVD, atherosclerotic cardiovascular disease. Herein, we review the mechanisms by which beta carotene and vitamin A control or modulate atherosclerotic cardiovascular disease. First, we summarize the current knowledge linking these nutrients with epidemiological studies or large group studies and lipoprotein metabolism as one of the initiating factors of atherosclerotic cardiovascular disease. So they're going to review the mechanisms by which beta carotene, which turns into vitamin A and vitamin A, control heart disease. Then they're going to link the nutrients with large-scale, large population studies and lipoprotein, that's the cholesterol metabolism, as one of the things that starts heart disease. They didn't leave much to discuss there. Okay, so let's go into this one. I don't know that we're going to go into too much detail on this one. I think we just go over the title. Early inhibition or stopping, slowing down, restricting, early restricting of endothelial, so the lining of the blood vessels is generally what they're talking about there, retinoid uptake, so the less vitamin A that is taken up by the blood vessels in the heart tissue upon myocardial infarction or after a heart attack, during a heart attack, restores cardiac function and prevents cell, tissue, and animal death. Wait, less vitamin A going into it. Less vitamin A going into the blood vessels in the heart restores heart function and prevents cell death and tissue death and animal death. Dang. That's a pretty good one, right? Don't want that happening. Now, I'm going to read through. This is Mike Roberto's post. He breaks this down really well, and I want to give him credit. So here, I'll put his link in the live chat if you want to go follow him on Twitter. He's doing amazing stuff. We actually chatted last night and just this morning about the malondialdehyde and LDL that we're going to go over today. So as he says, breaking, vitamin A deficient mice recover better from heart attacks. This post exposes some potential researcher lies too. Let's dig in. This study used LRAT mice, minus mice, which prevents normal vitamin A storage. So they don't store vitamin A well. That means it's going to be floating around more. They were fed a standard chow diet. Then at 18 to 21 weeks old, mice were split into two groups. 1, vitamin A sufficient. They gave them enough vitamin A. 2, vitamin A deficient. They didn't give them enough vitamin A. After 5 weeks on their updated diets, researchers measured serum retinol, blood vitamin A, cardiac function via echocardiography. You don't take any bile acids ever. That's not the fix to anything. Pumpster, we don't take bile to fix a bile problem. That would be like adding more water to your house that's flooding and thinking that if you add the right water, it's going to somehow fix the flooding. It doesn't work that way. Gene expression profiles. Okay, so then they induced a heart attack. I wonder how they did that, which toxin they used for that, and tracked recovery for 28 days. Results, post-heart attack. The vitamin A deficient mice demonstrated significantly better recovery, which makes sense based on that study we just mentioned, right? Getting their fractional shortening up to 18.9% in 28 days versus only 12.1% in the vitamin A mice. This was especially apparent on the males. More on this later. Oh, let me make this a little bigger. Okay. Now, here's where it gets dirty. In the abstract, they state, we found that uninjured vitamin A depleted hearts had decreased contractile function, but paradoxically improved recovery after injury. And he says, LOL, gotta love that word paradox. But is there ever really a paradox? I had to dig into fractional shortening to find out. Fractional shortening is the measure of the heart's contractile function, specifically how well the left ventricle, the heart's main pumping chamber, squeezes. Higher percentage means a stronger squeeze. Lower percentage means a weaker squeeze. Okay. Before heart attacks, at the baseline, the mice that got vitamin A had 54% fractional shortening. The vitamin A deficient mice had 46% fractional shortening. 46% is claimed to be worse than 54%, but they don't cite studies talking about fractional shortening. So is it? Take a guess. So Mike went digging into fractional shortening and found a bunch of studies that show that normal mouse fractional shortening percentage range is typically 35 to 45%. Values above 45% indicate hyperdynamic function, and values below 30 to 35% indicate dysfunction. This means that the vitamin A diet was basically giving these mice early signs of hypertension. They had high blood pressure. The low vitamin A diet almost brought them back into the healthy range. So remember, dysfunctional range, normal is 35 to 45. Dysfunctional is, above 45%. So the vitamin A mice already had dysfunctional heart function. And the vitamin A deficient mice were almost in the normal range. They probably had a high vitamin A diet, or the high vitamin A diet mouse chow before they were put on this program. So this low vitamin A diet was bringing them back towards normal. Okay, this makes sense. The body has to do less work to accomplish the same thing. You don't want your heart working too hard, but you also don't want it unable to contract. This is the Goldilocks effect. We don't want it too hot. We don't want it too cold. We want it just right. We don't want the heart working too hard. We don't want it like lazy and not doing its job. We want it just right. So not only does vitamin A impair a mouse's ability to recover from a heart attack, it seems to induce heart problems in the first place. There's even more. Liver health. The liver weight decreased by 20% in vitamin A deficient animals of both sexes. There were decreases in hepatic lipids, as in liver-based fats, in vitamin A deficiency. Literally everything is better without so-called vitamin A. So the liver weight going down is a good thing. Also interesting, in these mice, the females took way longer to deplete vitamin A stores. They didn't get the cardiac benefits from vitamin A deficiency because they hadn't gone deficient yet. And he says, makes sense, vitamin A is the one variable difference. Well, as he says here, unfortunately, this isn't any news to any of us in the community. Sorry, ladies, you have a longer road ahead. Ladies, you're better at storing fat-soluble toxins because you're better at making fat generally. That's why women are smoother than men. Ladies, you have a longer road to get better. The research shows it, and we've seen it. I'm sorry. I cannot change it. Of course, realize this is a mouse study and they're using genetically tampered mice to make it happen, but the data is absolutely wild, especially when you see that the abstract has what seems to be a bold-faced lie. Have a nice weekend, especially you, Chris. I'm assuming he means Master John here, which is a great little jab at a vitamin A addicted guy. It's time to dump the fat-soluble toxins. So that paper is right here. So here is, this is the paper. And I put this in the links. Vitamin A deficiency and male-specific effects on heart function in mice. So vitamin A I didn't see the lie in the abstract specifically that he mentioned, but it's there. Okay, so let's go into this. Retinol binding protein 4. So this is the protein your body makes to bind to retinol. So the only reason your body makes RBP4 is because there's retinol around. Your body makes RBP4 to bind to retinol to protect you from it. It's not some like Uber to take retinol around. It's to protect you from vitamin A in the blood especially. It's not it's a protective molecule. It's not some helpful, it's helpful because it protects you, but it's not like so nicely delivering vitamin A. If it's delivering vitamin A to any tissues, it's to store it there to get it out of your blood because you've got too much coming in. And your body has to constantly deliver it to places to store it. So again, retinol binding protein 4 promotes, makes worse, makes more cardiac injury after myocardial infarction, heart attack, via inducing cardiomyocyte pyroptosis through an interaction with NLRP3. Let's just go over the definition of pyroptosis so we all have that clear. A form of programmed cell death associated with antimicrobial responses during inflammation. A highly inflammatory form of lytic, that means cell destroying, programmed cell death that occurs most frequently upon infection with intracellular pathogens and is likely to form part of the antimicrobial response. So your cells are dying. Oh, I just got rid of the share. Sorry, Joe, I'm getting it. I thought I had it that time. Wait. I'll get it. Come on. Well, let me find it again. There we are. Okay, so here we are. Okay. So it's killing heart cells. Retinol binding protein 4 only exists when there's retinol around. It generally either has retinol in it or it doesn't. But it correlates closely enough that research continually says that you can measure RBP4 as a surrogate or a stand-in for serum retinol. So basically, the more retinol binding protein 4 you have, the more retinol you have around. That's it. Did I do anything in this one? Let me see if I had anything else on this one. Nope. Okay. So let's get into the next one. Serum levels of retinol binding protein 4 are associated with the presence and severity of coronary artery disease. So the more retinol basically you have in your blood, the more likely you are to have coronary artery disease or heart disease, and the more severe it is. It's pretty damning. So we have RBP4 is retinol binding protein 4 coronary artery disease. And then we have AMI, which is acute myocardial infarction. That's a heart attack. Stable angina, that's heart or chest pain, associated with the heart, but it's stable. Okay. Serum retinol binding protein 4 levels were significantly elevated in patients with coronary artery disease compared to non-coronary artery disease patients. Similarly, RBP4 concentrations increased with the number of coronary narrowed vessels. Do you remember how we talked about stenosis? The thing you guys need to know about blood vessel disease like atherosclerosis is that it's mostly calcium. The cholesterol was a red herring. Everybody's looking at cholesterol because they're like, cholesterol's in the plaques. They don't tell you that it's mostly calcium. How do we know that it's mostly calcium? When they measure your carotid for how narrowed it is, what do they call that? The CAC score. Coronary artery calcium. They measured the calcium in it to tell you how closed up it is. Didn't we just talk about vitamin A causing aortic valve calcification? It was shrinking the valve itself, and it was calcifying the valve flaps, the leaflets. It's shrinking your entire... Okay, when you think of a plumbing system, and there's limescale, hard water, what happens to those pipes and your fixtures and all that stuff over time? They calcify and they shrink. Same thing happens in your bloodstream. This is why we don't want to eat vitamin A. This is why we don't want to take vitamin D supplements, which cause the same thing. This is why we don't want to take tons of calcium supplements. Copper toxicity also calcifies things, which we'll go over copper in a second. Okay. Patients with coronary artery disease, both stable angina and acute myocardial infarction, showed elevated RBP4 serum levels. Notably, increased RBP4 concentration seemed to independently correlate with coronary artery disease severity. Oh gosh, this one, maybe we just need the title. Increased plasma retinol binding protein 4 levels in patients with inflammatory cardiomyopathy. So inflammation caused, so we have heart muscle disease caused by inflammation. Is it inflammation or is it just this little bad boy? This was an interesting thing. I thought that the liver made retinol binding protein mostly, and I'm not sure if this is correct, but anyway, they say here retinol binding protein 4 is an adipose tissue derived protein, which is weird because how does fat make proteins? Anyway, it's just interesting. I thought the liver made it because that's everything I'd read before was talking about the liver making retinol binding protein. But apparently your body fat does too. Maybe both of them make it. The liver gets fatty liver, right? Body fat makes stuff to protect itself. Again, I mean, how would your body fat and your liver transport retinol binding protein or retinol back and forth? It would bind them to this protein to protect you. Right? Why would body fat need to make vitamin A binding proteins if they store it there? Well, it's just to send it back to the liver with a bodyguard, an escort, so it doesn't hurt everything on the way back. And then they're saying, so the retinol binding protein has pro-diabetes causing effects. Let me see if there was anything else here. I think that was it. Okay, another one. This one's great. Hey, do you know they can cause heart failure by giving vitamin A? A novel mouse model of heart failure with preserved ejection fraction after chronic kidney disease induced by retinol through the JAK-STAT pathway. So they give them chronic kidney disease induced by retinol. So what did Grant fix by going on a low vitamin A diet? Oh, he fixed chronic kidney disease completely. So they gave these mice chronic kidney disease with vitamin A, and then they created a new model of heart failure. Gosh, what would build up in the blood if you couldn't clear it through the liver and the bile? I mean, it's the liver and the kidneys. You'd build up retinol in the blood, and it causes damage to all of it. So what's the first thing we do? We eat less of it. Heart failure with preserved ejection fraction is the most common type of heart failure in chronic kidney disease patients. Based on these features, we established the practical that was heart failure with preserved ejection fraction after the chronic kidney disease mouse model using 5/6 subtotal nephrectomy and retinol administration. Serum accumulation of retinol. Blood vitamin A going higher in chronic kidney disease prompts myocardial hypertrophy, the growth of the heart muscle in a bad way, and fibrosis, that's the scarring of the heart muscle in a bad way, by activating these things. Ooh, it's not looking good for you, vitamin A. Okay, let's keep going. Now we get into copper. I'm not going to spend a lot of time on this. Copper is a toxic metal, folks. If you want to think it's good for you, go ahead. FAFO. Go ahead. Drink out of your copper cups, drink out of your copper water bottles. Come back and find me when you realize you messed up and you found out. Circulating copper levels and the risk of cardiocerebrovascular diseases. So we have heart and brain blood vessel diseases and cardiovascular and all-cause mortality. The risk of death from all causes and from heart disease. A systematic review and meta-analysis of longitudinal studies. That generally is going to mean longer studies. Elevated circulating copper levels were linked to the risk of stroke, coronary artery disease mortality, cardiovascular mortality, and all-cause mortality. Wait, so I just want to make sure the higher people's copper levels were, the higher their risk of stroke, the higher their risk of coronary artery disease, the higher their risk of dying from heart disease, and the higher their risk of dying from all causes. Continuous analysis. So they're going as the copper is increasing in the blood. Showed consistent results regarding the association between copper levels and stroke incidence, cardiovascular mortality, and all-cause mortality. So the higher the copper went, the more risk of all those things. Excessive circulating copper levels are associated with stroke, coronary artery disease mortality, cardiovascular mortality, and all-cause mortality with moderate certainty. Well, if they're at moderate certainty, you can be sure it's real. Association of serum copper with cardiovascular mortality and all-cause mortality in a general population, a prospective cohort study. Maybe this study was included in that other one. Maybe it wasn't. The present study suggests excessive serum copper concentrations are associated with the risk of cardiovascular disease and all-cause mortality in American adults. Let's keep going. Serum copper levels and the risk of major adverse cardiovascular events, a systematic review and meta-analysis. Now, I'm telling you, copper toxicity or too much copper for you. This does not mean like the LabCorp ranges for serum copper are obscenely high as the reference range. I think it goes up to like 160 or something. It's obscene. It shows how toxic in copper everybody is because they only take out the top 2.5% of either side of all the people they test. So that means they're going to say 95% of people are normal by definition. So only 2.5% of people are higher than like 166, I think it is. Folks, you can have copper. I want copper on serum below a 90. And the reference range is up to 166, I think, if I recall correctly. The entire population is horribly copper toxic. Yeah, as Will is saying, great news, Mom. The researchers said I'm normal. No, Normal copper levels are disgustingly high. They're allowed to be high. When I get people at like 120, oh, they're in rough shape from copper. They are in rough shape. Imagine 160. So let's see what we got here. High serum copper levels. Notice they didn't, well, anyway, high serum copper levels were associated with an increased risk of total stroke, myocardial infarction, heart attack, and cardiovascular mortality, the risk of death from heart disease. Hybrid studies seems to modify the strength of the association between serum copper and the risk of cardiovascular mortality and ischemic stroke. So basically an infarction or a blockage of blood vessel to your brain. Yeah, don't. Now, I don't want to spend too much time on ferritin. Ferritin, many people already understand that high ferritin or high iron is associated with heart disease. So I'm going to go through this pretty quick. Elevated ferritin levels have been reported as a risk factor for coronary heart disease in Finnish and Italian studies. I want you to understand, like LabCorp's ferritin, high ferritin on their lab in the reference range is 400. That's obscene. That's obscenely high. So when they say elevated, they mean like way elevated for where I want people. So C-reactive protein is CRP. So what do we have here? In women, serum ferritin correlated with, I want you to pay attention here, folks. This is going to explain overtypes. Overtypes I talk about in the Love Your Liver program. In women, serum ferritin going up or going down correlated with CRP, C-reactive protein, a marker of inflammation, waist measurement. So what that means is the ferritin went up with the waist size. Body mass index, a measure of a general, population-wide sloppy measure of how fat somebody is versus how tall they are. And triglycerides. So all of these things, they're saying serum ferritin went up as CRP went up, as waist measurement went up, as body mass index went up, and as triglycerides went up. Triglycerides are directly related to vitamin A toxicity, if you didn't know. In multiple regression analysis, CRP alone was independently associated with serum ferritin. So inflammation marker independently associated with a higher serum ferritin. Serum ferritin in men correlated with, so higher serum ferritin in men correlated with a bigger waist, higher BMI, higher triglycerides, and higher high-density lipoprotein cholesterol. Okay, you got one W in there. After adjustment for the other variables, waist measurement was the only independent predictor of ferritin. So they're saying they could measure people's waist size and predict ferritin decently well. Ferritin levels in young men and women are associated with obesity and serum triglycerides, HDL cholesterol in men, and inflammation in women. Overtypes. What do I talk about with overtypes in the program? Generally, overweight, higher lab numbers, especially on blood tests. So higher, they tend towards higher ferritin, higher copper, higher vitamin A. Undertypes are the opposite. It's just a trend. It's just a pattern. The association of ferritin with, and remember, so I have a whole episode. Joe, you may want to put this up. I have a whole episode on ferritin and how that correlates with toxic bile paradigm. I don't know which number it is, but Joe, you could bring that up for people. The association of ferritin with cardiovascular, heart disease, and all-cause mortality in community dwellers, the English longitudinal study of aging. Circulating ferritin levels showed sex-specific prognostic patterns. So there were patterns that were associated with gender, okay? Or sex, whichever one. They're the same thing. High ferritin levels in men with no major chronic disease. So if somebody just has high ferritin and no other major diseases, and low ferritin levels in all women were associated with increased all-cause mortality after adjusting for covariates. High ferritin levels in men with no major chronic diseases were also independently associated with an increased risk of cardiovascular mortality. So this is like, possibly like as I've talked about before, the wrestling coach at my high school. I didn't do wrestling, but he was the wrestling coach at my high school. He was also a PE teacher. This would be like, okay, so they have no major chronic diseases. This is like, yeah, the wrestling coach at the high school, so healthy, man. He goes jogging every day and he looks fit and all this stuff. And then he croaked one day running around the park track. He died of a heart attack. High ferritin levels in men with no major chronic diseases were also independently associated with an increased risk of cardiovascular mortality or the potentially croaking of a heart attack while out jogging. Okay? Let me see if I have anything else on this one because I want to go over the men and women thing here. Okay. Folks, as I go over in the ferritin episode, high ferritin is associated with liver problems, toxic bile leaking. Ladies, if you have chronically low ferritin and your doctor's always telling you you're anemic and there's no good reason for that. So if you, so an example, if you are bleeding heavy every month, like bleeding heavy every month will drain you of blood and you can have low iron and low ferritin in the blood. What do I tend to look at for that in terms of what would I focus on in that? Well, copper toxicity would be huge. So would vitamin A toxicity. So we'd want to fix those because that's what's causing the heavy bleeding and zinc deficiency and all the other things that we're going to talk about today. Now, if you are chronically low ferritin and you don't bleed heavy and you eat meat, red meat, like at least once a day, if you eat red meat once a day, you don't have an iron deficiency in your diet. Okay, so if you don't have if somebody was a vegan and they're like, I have low ferritin, I'd be like, of course. Yeah, that makes sense. So somebody who eats red meat once a day at least and does not have heavy bleeding monthly and they chronically have low ferritin or anemia, I'm going to tell you folks, and then maybe you've taken iron injections. Maybe you've taken tons of iron pills and it barely brings up your blood markers. And as soon as you stop taking them, they drop again. You have a liver problem. We fix this. Your chronic anemia the problem is in your liver. So when we say liver problems are associated with chronic disease, low ferritin levels in all women were associated with increased all-cause mortality, death, risk of death from any cause. Well, if you had liver problems at the root of all of it, would it increase your risk of death from any cause? Yes. This is what we talk about. Men tend to go high on iron. So like there's some patterns that we can see. These are not, again, these are patterns. These are trends. Men tend to get diarrhea. Women tend to get constipation. Men tend to get high ferritin. Women would tend to get low ferritin much more commonly. I'm not saying it's common, but I'm saying much more commonly than men. There's just these patterns. The sex hormones and the ability to make bile and process toxins has a very distinct effect on different genders, okay? And no, whatever you identify with doesn't change that risk. Ferritin, inflammation, and iron deficiency in acute heart failure, evidence from the Edifica cohort. Look at this. The median ferritin plasma concentration at admission was 180 picograms per mil. Yeah, that's high. Patients with higher ferritin levels at admission, so they're even higher than that, were predominantly men. What did I just say? Men tend to have higher ferritin. Men can handle higher ferritin better than women. And what I mean by that is a man with a 200 ferritin is generally handling that ferritin better than a woman at 200 ferritin. It doesn't mean it's good for either of them. Women tend to tolerate higher copper than men. That doesn't mean it's good for them. They were predominantly men exhibiting a high prevalence of chronic kidney disease and alcohol consumption. Poisons beget poisons. The more toxic somebody is, the more toxins they want to consume. And presenting with lower blood pressure and a higher incidence of clinical infection. Higher ferritin levels were associated with an increased risk of the composite of heart failure hospitalization or cardiovascular death. Oh, Dylan, we're only halfway through. Dylan was saying, I love how other people go through like 2 studies and I think it proves their theory while Dr. Smith does like 20. Yeah, I think we have probably about 40 papers to go through today. I'll look and see. I'll get an estimate of where we're at. Let me see if there was any other ones here in there. Elevated ferritin levels are independently, by themselves, associated with poor prognosis or predictive. They're saying you're not going to do well. They're predicting that you're not going to do well in acute heart failure. Okay, let me get back up here. Ferritin levels and risk of heart failure, the atherosclerosis risk in communities study. Oh yeah, HF is heart failure. Derangements in iron metabolism. So your iron metabolism is not good. This could be a problem of the liver and you have high ferritin, or you could have low ferritin, which we were just going over. Either low or high ferritin serum levels were associated with a higher risk of incident heart failure in a general population, even without concurrent anemia. These findings suggest that iron imbalance might play a role in the development of heart failure. Gosh, you think? Oh, one of the things you need to know about iron, folks, and do not use this if you have low ferritin, ladies, but it's funny, there's a group out there that follow a protocol who obsess over iron overload. They obsess over it. They go and donate blood even though they don't know their ferritin, and they'll continue donating until they drive themselves anemic, and then when they're anemic and they feel like crap, they get told that they're just detoxing iron and they should keep donating iron or keep donating blood. It's delusional. They also talk a lot about bioavailable copper, which is pseudoscientific nonsense. Anyway, they love their vitamin A. They'll do cod liver oil. They'll eat lots of liver and eggs and all this. They eat tons of vitamin A. Vitamin A increases your absorption of iron. So they obsess over iron overload, and yet they eat tons of vitamin A on purpose, and then they wonder why they're obsessing over iron overload. And then they also make themselves copper toxic. So pretty fun. When we get those people, yeah, they're pretty messed up too. Oh, wait, did I finish that one? No, I did. Okay, so let's go here. This was... yeah, a comprehensive review on serum ferritin as a prognostic or a predictive marker in intensive care units. So this is somebody who's already in intensive care. So these things would be indicators of, are they bad in real life too? Into ischemic heart disease, that's lack of oxygen, lack of blood flow. Serum ferritin has garnered considerable attention as a prognostic or a predictive marker in intensive care units. So they can tell you how bad you're probably going to do if you go into the intensive care based on your ferritin, is what they're saying. IHD is ischemic heart disease. Elevated serum ferritin levels have consistently been associated with adverse outcomes in ICU settings, including increased mortality, more death, prolonged hospital stays, poor recovery, and higher morbidity rates, getting other problems along the way. Furthermore, the relationship between serum ferritin levels and ischemic heart disease, which heart attack would be a form of ischemic heart disease. You're blocking off blood flow and oxygen and the tissue dies, and so would a stroke, underscores its potential as a biomarker or a way to predict for cardiovascular risk assessment in critically ill populations. Well, how did they get to be critically ill, folks, right? We got to think about that. Now, oh, this is just an extra one. Some people are out there, mineral chiefs, talking about they love their manganese and they're taking lots of manganese. I just threw this one in there. Manganese also causes cholestasis or toxic leaky bile. Cardiovascular toxicities upon manganese exposure. Pay attention here, folks. Manganese-induced Parkinsonism. Parkinson's disease has been well documented to be connected to manganese exposure. Well documented. And there's people out there trying to say it's an essential mineral that you need. Have fun with that. However, little attention has been devoted to manganese-induced cardiovascular dysfunction. Also, cholestasis and liver problems. Animal studies indicate that, pay attention here, manganese is capable of quickly accumulating in heart tissue. Quickly accumulating in heart tissue. Resulting in acute or subacute cardiovascular disorders such as acute cardio depression and hypotension, low blood pressure. So possibly out there, if you're out there and you have low blood pressure and you can look back on your history and you've taken a lot of manganese, and I would include multivitamin manganese in this, that might be part of it. These toxic outcomes appear to be associated with manganese-induced mitochondrial damage. Pay attention, mitochondria nerds, and interaction with the calcium channel in the cardiovascular system. So there's that one. Oh, now we get to go into the fun part, our speciality. The role of aldehyde dehydrogenase 2 in cardiovascular disease. What is aldehyde dehydrogenase, folks? It is the enzyme that detoxes aldehydes. Okay? Sorry, Joe, I'm going to try. I'm making sure I'm centered so I can stay. I keep moving my mic. I tend to, anyway, I do that. No one said I was a professional at TV or whatever you want to call it these days. Anyway, the role of aldehyde dehydrogenase 2. Okay, so one of the problems with your health, folks, is you are building up aldehydes in your system, because many of the things we have all done in the past we're taking in too many aldehydes, and we're doing things like supplements or medications that slow down your aldehyde dehydrogenase so you don't process the aldehydes and then they build up in your system. Aldehydes are toxic by definition. Gosh, wouldn't that be weird if we say vitamin A is a toxin and vitamin A turns into retinaldehyde? And then if you block ALDH enough, you don't process the retinaldehyde and it doesn't leave your system and it accumulates in your liver and other tissues because it can turn back into retinol. And then retinol binds to a fatty acid ester and it can be stored in your liver or in your body fat. And then we see all those retinol binding protein things, and then it's adipose tissue making retinol binding protein. Fat soluble toxins store fat storable toxins. Fat soluble vitamins, very commonly A, D, and E, are stored as fat soluble toxins. Don't take rat poison vitamin D3. Get sunlight, and you get your zinc and get your magnesium and get your muscle meat protein. And that will help fix your vitamin D. Even if you need to get a Spurdy, the link, affiliate links down in the info there if you want to get a Spurdy lamp because you're somewhere where you don't get sun, or if you want to join a gym or a tanning salon to get some tanning bed time in the winter, you'll feel better. You'll be happier. You don't have to do a lot of it. You just need to do some. Actually, I went over it. I went over a lot about tanning beds in the live stream podcast I was on yesterday, the Missing Link podcast. If Beth, if you're here, if you could post that link, or if anybody else has that Missing Link podcast that I did yesterday, if you wanted to put it up for other people to see it here, that'd be great. So the role of aldehyde dehydrogenase 2 in cardiovascular disease. Aldehyde dehydrogenase 2 is a mitochondrial. You know, the funny thing is I don't ever talk about mitochondria because I don't need to, because all the things that we do fix the mitochondria stuff. I don't obsess on mitochondria. We obsess on the outcome. Is a mitochondrial enzyme involved in the detoxification of alcohol-derived acetaldehyde. It also detoxes retinaldehyde and formaldehyde and endogenous, wait, pay attention here, endogenous aldehydes made inside your body. Retinaldehyde would be one of those because it starts as either beta carotene or retinol. The inactivating ALDH2 RS671 polymorphism. What they're saying is that there is a genetic component where people can make less ALDH than others. They are behind the 8 ball to begin with. It is present in up to 8% of the global population and up to 50% of the East Asian population. So if you are Asian and you are listening to this, there's a 50% chance that you have slow ALDH. Slowed. Doesn't mean it doesn't function, it just means it's slowed. If you are of any race and you get what's called the Asian flush or the Asian glow, or you call yourself allergic to alcohol because when you eat it, you tend to get flushed and you tend to get intoxicated very easily and you tend to have bad hangovers, you have slow ALDH. It could be genetic. You could have caused it with the things you've done in your life. Aldehydes are toxic. Retinaldehyde is toxic. So, slow ALDH genetically is associated with increased risk of cardiovascular conditions, heart conditions such as coronary artery disease, alcohol-induced cardiac dysfunction. So they're more prone to having alcohol problems, pulmonary arterial hypertension, heart failure, and drug-induced cardiotoxicity. Many drugs inhibit ALDH. Looking at you, ivermectin and fenbendazole, methylene blue, quercetin, all these things that they're pushing for long COVID and fixing your jab injuries, they all slow ALDH. Oh, what's another way of making people infertile? The male contraceptive pill, specifically. One of the reasons it hasn't come out yet is because it's so toxic, because the mechanism of it is it slows down ALDH, so you build up aldehydes, and the aldehydes ruin your testicles, guys. This is all part of the grand plan. Although numerous studies have attributed an accumulation of aldehydes secondary to alcohol consumption, that's one way you can get it, ischemia, lack of blood flow is one way you can get it, or elevated oxidative stress to an increased risk of cardiovascular disease. This accumulation alone does not explain the emerging protective role of this genetic polymorphism against aging-related cardiac dysfunction and the development of aortic aneurysm or dissection. So they're saying this ALDH by itself, this ALDH genetic component, doesn't explain the fact that they're linking aging-related cardiac dysfunction and aortic aneurysms to this same genetic difference. ALDH2 can change, modulate, or control risk factors associated with atherosclerosis, such as cholesterol biosynthesis and HDL biogenesis, your ability to make HDL in liver cells, and foam cell formation and efferocytosis in macrophages via non-enzymatic pathways. Hold on, I don't know what efferocytosis is. I've never heard of that. In cell biology, efferocytosis is the process by which apoptotic cells, programmed cell death, so they're dying, they're killing themselves, suicidal, are removed by phagocytic cells, like white blood cells. It can be regarded as the burying of dead cells. So if it's increasing efferocytosis, what does that likely mean is happening to cells? They're dying. The aldehydes are killing them. Okay, so let's keep going. You want your ALDH to be running well, and we are the experts in helping to fix it. You do not take things like dihydromycin or DHM to accelerate your ALDH. It is a no-go. It's a non-starter. It will cause more problems than it solves. Don't go out and try to accelerate your ALDH. Try to stop putting aldehydes into it and try to take the right nutrients that we cover here, and we cover in like keystone minerals that give you the nutrients you need to run ALDH properly. It'd be like, as an example, a lot of people out there, like you'll hear about ALDA1, which is an ALDH accelerator. It's a pharmaceutical ALDH accelerator. Taking a pharmaceutical ALDH accelerator or trying to take dihydromycin is like trying to put nitro in a car engine when there's just not enough gas. You need the gas. You need the gas first. You don't need the nitro unless you have the gas and the oil. You need the fluids. You need to run the engine first before you try to jack it up. And then when you find that you jack it up, you just might drive around the curve too fast and kill yourself because you can't control the car anymore. Here we go. What was this one? Aldehyde dehydrogenase 2 roles in ischemic cardiovascular disease. Accumulating data. More and more data showing up have shown that ALDH2 can help restore mitochondria function by eliminating toxic aldehyde and participating in cellular signaling important for cell adaptation and survival. Well, what's the toxin we're talking about here when we're talking about cell signaling? We're talking about aldehydes, right? Because they're going to try to, I'm sure at some point they'll try to say that aldehydes are some sort of magical cell signaling, like retinaldehyde. They'll try to say that toxic aldehydes are necessary for cellular signaling. This is the gaslighting that they do. So they can keep having their research money come in. Okay. So what happens when they have these genetic slow ALDH? Oh, it's associated with an increased risk of early onset coronary artery stenosis. The tubes are shrinking because of calcium deposits. Wait, let me find what I wanted in here. In men less than 55 years old and women less than 65 years old, individuals with ALDH2, the slow ALDH genetics, overweight, overtype, smoking history, smoking and nicotine slow down your ALDH. That's all part of the infertility psyop too. And diabetes mellitus. You know how you get diabetes is you eat too much vitamin A. Increase the risk of developing, I believe that's coronary artery syndrome. Yeah, coronary artery stenosis. No, so that's coronary artery stenosis. So aldehydes are connected to calcifying your blood vessels short, or calcifying your blood vessels smaller as well. Sounds like heart disease to me. Okay, aldehyde dehydrogenase 2 activation in heart failure restores mitochondrial function and improves ventricular function and remodeling. So when they activate it and they start breaking down the aldehydes, it restores mitochondrial function and improves the remodeling and the function of the heart. Gosh, do you wonder why when they restricted vitamin A from those mice and they recovered better after the heart attack they induced? Well, they could detox the aldehydes better too. One of the aldehydes is retinaldehyde. Funny, it all connects. So they gave the ALDA1. Yeah, ALDA1. Selective activation of mitochondrial ALDH2 is sufficient to improve the heart failure outcome by reducing the toxic effects of aldehydic overload on mitochondrial bioenergetics and reactive oxygen species generation, suggesting that ALDH2 activators such as ALDA1 have a potential therapeutic value for treating heart failure patients. How about this first? We stop putting in the aldehydes and we stop taking drugs and supplements that slow down your ALDH. And then we give your body the nutrients that it needs to run ALDH instead of taking pharmaceutical meds. Does pharma want us to do that? Nah, they don't want us to do that. But they don't sell ALDA1. You can't get ALDA1 as a medication anywhere. You might be able to find it on some research chemical websites. If you decide to play around with that, don't come to me telling me you messed yourself up. Don't even come to me and saying you did that stupid crap and what do you do now? I don't know how to fix you from it. You took a pharmaceutical. You did something. You took a toxin that had an effect. Then we just do what we normally do for you. You'll be back to square one. Those of you who decide to do wild experiments, especially when I've told you not to, and you go out and you damage yourself, do not think that I have magical things up my sleeve to fix your dumb maneuver. We'll do the same things that we did. I fix Accutane by doing the same things. I fix Accutane damage by doing the same thing. So if you go out and you do something too much or the wrong thing, and I warned you not to, you're just going to be back to doing the same stuff that you were doing before. But you'll have set yourself back because now you have more damage to undo. Serum malondialdehyde. So malondialdehyde. Okay. What is one of the bad things, or two of the bad things about seed oils? Well, there's multiple bad things about seed oils. First, why are seed oils generally yellow? Is yellow a marker of a toxic color in nature? Aposematism, that kind of thing? Yes. Seed oils that are yellow are full of toxic vitamin A, carotenoids, okay? And other yellow bitter things. PUFAs, the polyunsaturated fatty acids that tend to be higher in seed oils. They're not always the highest in seed oils, but they tend to be higher in seed oils. When those go into your body and they oxidize, they turn into malondialdehyde and 4-hydroxynonenaldehyde. So eating an excess of PUFAs will generally create an excess of malondialdehyde and what's called 4-HNE for short in your system. These are bad. These are associated. You can probably look up almost any disease you want to look up and find that malondialdehyde levels are higher in that disease. So if you eat less, so first of all, if you're taking seed oils and they're yellow, you're taking in carotenoids, vitamin A, which are going to turn into retinaldehyde. Then you're taking in PUFAs, which are going to turn into malondialdehyde and 4-hydroxynonenaldehyde. So now you've got at least 3 aldehydes coming into your system. Now, we have research showing that malondialdehyde itself inhibits ALDH. It slows down your ALDH. So the very toxin that you just created in your system slows down the detox pathway of it. How vicious cycle is that? So what do we do? So you can, okay, every natural fat that you can eat, folks, is going to have some PUFAs in it. So the more fat you eat of any type, the more PUFAs you are ingesting. Doesn't mean all of them are high in PUFAs. It just means, like, even if you're eating coconut oil, you can go look up coconut oil and there's PUFAs in it. You can go look up tallow and there's PUFAs in it. So the more of any of these fats you eat, the more PUFAs you're eating and the more malondialdehyde and 4-HNE you're making. And the more fat you're providing to your body to increase your absorption of those fats and the fat-soluble toxins, and the more lipophilic bile acids you're going to make in your gut biome, which help you to absorb all that stuff, and they're more toxic bile acids. So no, eating more fat is not the answer. Okay, serum, and I want you to pay attention here because I sent Mike this study earlier this morning. Serum malondialdehyde dash modified low-density lipoprotein levels on admission to, I'm guessing, the hospital, predict prognosis. They predict the prognosis. Okay, that seems like kind of redundant, but they predict the prognosis in patients with acute coronary syndrome undergoing percutaneous through the skin coronary intervention. Did they just say that your body binds malondialdehyde to LDL? Pretty sure that's what they just said. LDL is considered the bad cholesterol, right? Or lousy. LDL, lousy cholesterol. Wait, it actually binds to a toxic aldehyde? What does your body make LDL for? It's a marker of how much toxicity you have, and your body binds it to toxins. So here's the problem. Here's where people think that higher cholesterol is a good thing. If you are really toxic, you have to follow me here. If you are really toxic, you want to make a lot of LDL to bind to the lot of toxicity that you have. You don't want to have a lot of toxicity and a little LDL. Does that make sense? If you're going to be really toxic, you want a lot of LDL to bind to it so that you're protecting yourself at least as much as you can. It'd be like if you have a lot of retinol, but you're not making enough retinol binding protein, free retinol floating around your system is really bad. So you want to at least be able to make enough retinol binding protein to bind to it, even though the retinol binding protein by itself might be bad. What we don't want is, like, so if when they say that, oh, low cholesterol could be just as bad or worse, low cholesterol and lots of toxicity is worse. This is why, like, if somebody always had low cholesterol, but they're really toxic, as we start getting them better and their liver can start doing what it's supposed to do to protect them, it might actually make more cholesterol for a while. Your liver makes 80% of your cholesterol. Why people think you have to eat it when your liver can make it is pretty humorous to me. So, malondialdehyde modified low-density lipoprotein, MDA-LDL, is a predictive marker of cardiovascular events in patients with stable angina pectoris. This is the chest pain that's stable. Heart pain, chest pain. Okay? MDA-LDL levels, so the level of malondialdehyde bound to LDL, this combo, in patients with acute heart attacks were significantly greater than those in patients with unstable angina pectoris. During follow-up, 82 events occurred. 22% of the people had an event. Kaplan-Meier analysis showed that patients in the highest MDA-LDL tertile, the highest third, so there'd be there'd be 3 divisions. The highest division had the worst prognosis. They were predicted to have the worst outcome. Cox regression analysis showed that blood MDA-LDL levels were an independent, all by itself, predictor of cardiovascular events, heart disease, after PCI was what? What was PCI? Percutaneous. Oh, so this is after percutaneous coronary intervention. This is probably where they just went into their blood vessel through where they go in stents and stuff like that. Okay. After percutaneous coronary intervention in patients with acute coronary syndrome. Is that ACS? Yeah, acute. Get out of here. Acute coronary syndrome, yes. Just want to make sure these abbreviations are right because they'll use different abbreviations for different things or the same abbreviations for different things. It was an independent predictor of cardiovascular events even after adjustment for age, sex, body mass index, conventional cardiovascular risk factors, other lipid biomarkers, statin use on admission, cardiac biomarkers, and presence or absence of multivessel disease. Why aren't your doctors measuring serum MDA-LDL? If it's an independent predictor by itself, even after adjustments for age, sex, how fat you are, conventional risk factors, other lipid biomarkers, which would be cholesterol, HDL, LDL, triglycerides, that stuff, whether you're using statins or not, cardiac biomarkers, maybe they mean CRP or something like that, or homocysteine, and presence or absence of multivessel disease, where they could actually tell by looking in your blood vessels whether you have problems or not. Why isn't anyone measuring this? Serum malondialdehyde and prevalent cardiovascular disease and hemodialysis. So remember, malondialdehyde goes through ALDH, but malondialdehyde itself will slow down ALDH. It's a bad combo. CVDs, cardiovascular disease, hemodialysis. So these are people who have chronic kidney disease and they have to go in for dialysis to filter their blood because their kidneys suck at it because they're damaged and toxic and deficient. And then malondialdehyde is MDA. That's a pretty common abbreviation, MDA. Mean serum MDA levels, malondialdehyde levels, were significantly elevated in, what was HD again? Hemodialysis patients. So chronic kidney disease people had more of this. You remember how I told you any disease you look up, they're going to have more malondialdehyde levels. In hemodialysis patients with prevalent cardiovascular disease compared to those without. So they mean there was more malondialdehyde in the hemodialysis patients who had heart disease than those who were also hemodialysis patients but didn't have heart disease. Patients in the highest compared with the lowest tertile of post-dialysis malondialdehyde. So they measured them after dialysis. So the people who still had the highest malondialdehyde compared to the lowest were nearly 4 times as likely to have prevalent cardiovascular disease. And serum malondialdehyde was the single strongest predictor of prevalent cardiovascular disease in this patient population. You don't want malondialdehyde. So what do we do? So think of the 3 basic approaches that we do here to restore people's health. 1, we reduce or minimize or eliminate the sources of the toxins coming into you. 2, we facilitate the removal of stored toxins or of toxins that are in you. 3, we provide the body with the nutrients that it needs to run detox and to protect yourself against it. So what do we do with aldehydes? We reduce the sources of aldehydes, including vitamin A. We help the body to get rid of it. Well, what are we trying to do? We're not trying to speed up ALDH. I said that wrong. We are trying to restore ALDH to its proper function because the ALDH is what processes the aldehydes. Number 3 is we give the body the nutrients that it needs to help protect itself from aldehydes and to run ALDH. Those 3 main ones being selenium, molybdenum, and nicotinic acid, aka flush niacin. That's how we fix this problem. And how do we know that it works? Well, one of the ways we know that we are restoring aldehyde dehydrogenase properly, I'm not suggesting anybody drink alcohol. but people on the program are still going to decide to imbibe on occasion. Well, they say they hardly get a buzz anymore and they don't get hangovers because their livers are processing the alcohol so fast. Now, you can ruin that. You could drink a bunch of alcohol and put a bunch of aldehydes into your system and use up your ALDH and set yourself back. For those of you who are choosing to go out and drink and you're finding that it's really hard to even get a buzz, you're just wasting your money on alcohol, aren't you? And you're just adding toxicity to your system. I mean, if you just want to do it to fit in and have a drink or two, that's one thing. But if you're going and trying to get a buzz when your liver's working really, really well, and now you're just having to pay a whole bunch more money to hardly get a buzz, it seems like kind of a bad deal. But yeah, this is how we know people's ALDH is working better, is because when they put in a source of a lot of acetaldehyde, which is alcohol, it doesn't affect them as much. Hangovers are acetaldehyde. Okay, let's keep going. The nutrients, I don't want to spend. Oh, thank you, Hound Jog. So let's go here. Let's go this one. Yes. Okay, magnesium. Magnesium is kind of like ferritin. If you guys have been in health at all for any amount of time, you should know that magnesium is critical to your health. Critical. Magnesium and heart disease are one of the most obvious connections. If you want to watch my topical magnesium episode on the channel, I would do that because I go over all the different topical magnesium approaches that we use. No, we don't use Epsom salts. No, we don't use magnesium sulfate. That's the sulfate slows detox. We use magnesium chloride. If you want to see my magnesium lotion, which is a huge favorite among people here, works really well. 2 pumps a day will get you what you need. But yeah, topical magnesium, in my opinion, is 10 times as effective as pills. Can you use pills of magnesium? It's not going to raise your systemic magnesium very well. It will help you poop, but it's not going to work very well to raise your systemic magnesium. 2 pumps a day of my magnesium lotion. Generally, your magnesium levels are going to be covered for most people. That's it. Okay. Magnesium and cardiovascular disease. Well, magnesium is the most abundant intracellular, that's inside the cell, divalent cation. That means it has a charge of +2. And essential for maintaining normal cellular physiology and metabolism. So if it's essential for normal function, it's pretty important, right? It's essential. Acting as a cofactor of numerous enzymes, regulating ion channels and energy generation. In the heart, magnesium plays a key role in modulating neuronal excitation, that's the nerves, intracardiac conduction, conducting the electrical flow between parts of the heart, and myocardial heart muscle contraction by regulating a number of ion transporters, including potassium channels and calcium channels. Magnesium also has a role in regulating vascular tone. That's how tight your blood vessel muscles are contracting or not. Atherosclerosis and thrombosis, these are clots. This is blood vessel disease, basically. Vascular calcification, that's stenosis. And proliferation and migration of endothelial and vascular smooth muscle cells. As such, magnesium potentially has a major influence on the pathogenesis, disease creation of heart disease. So do you want to see here? So we're talking about an essential mineral. And then they say it has a major influence on the creation of cardiovascular disease. Do you see how they just badmouthed an essential mineral? Whereas before they were talking about vitamin A and they're like, or bile, and they're like, it's critical to cardiovascular health. Do you see how they did the opposite? They are gaslighting you. Magnesium deficiency, potentially. Why did they not add deficiency? Potentially has a major influence on the disease creation of cardiovascular disease. Okay. Observational data has shown an association between low serum magnesium concentrations. So low blood magnesium is connected to increased atherosclerosis, coronary artery disease, arrhythmias, heart rhythm problems, and heart failure. Okay. Or magnesium intake. So if people have low magnesium intake, so they either have low blood magnesium or they have low magnesium intake, and they also see increased atherosclerosis, coronary artery disease, arrhythmias, and heart failure. And then, so this is what they do, because this is pharma medicine. As such, there's currently no firm recommendation for routine magnesium supplementation, except when they've diagnosed it in the blood as a cause for cardiac arrhythmia. So you have to have an irregular heartbeat. Your doctor has to order the magnesium blood test, and then you have to be actually low, which means you're in the lowest 2.5% of the people who have gotten that blood test at the lab before they'll say that you should take magnesium supplements. They don't care about you. What's this? Magnesium for the prevention and treatment of cardiovascular disease. In industrialized Western countries, a low intake of magnesium often predisposes or makes someone more easily getting this to a high prevalence of magnesium deficiency, right? If you don't take in enough magnesium, you're more likely to have a magnesium deficiency, duh. Increasing the risk of cardiovascular events and cardiovascular death. So death risk from heart disease. Let me go find the other spots in here. In recent studies of hospitalized patients, 42% were shown to be hypomagnesiemic. Okay, do you understand what I just said? So that means they're low in the blood test, which means they're in the lowest 2.5%. 42% of the people in the hospital were in the lowest 2.5%. Pretty important. However, physicians, the experts that they are in health, right, request magnesium testing in only 7% of these patients. So wait, let me get this straight. So doctors only ordered 7% of the patients in the hospital. They ordered a blood magnesium. Only 7% of them. 42% of that 7%. So that's like what? 3% of all the people that, I mean, I'm sorry, I won't do that math in my head. They only ordered it on 7% of the people. And in 7% of those, 42% of them were so low in the blood that they were in the lowest 2.5% of people who had gotten their blood magnesium measured. If you don't understand how bad that is, that's really, really bad. That means there's tons of magnesium deficiency out there. And second of all, blood tests for magnesium suck, as we'll go over. In a study conducted among patients in the intensive cardiac care unit, so heart care unit, 53% of the patients had mononuclear cell magnesium content below the lowest normal control. I'm not sure if this is white blood cell magnesium or red blood cell magnesium. But anyway, half of them getting heart hospital care, more than half of them were below the lowest normal control person who was probably deficient also. Clinically, serum magnesium is usually measured despite the fact that less than 1% of magnesium exists extracellularly. So they're measuring serum magnesium. They measure serum magnesium. I just told you that it sucks. Only 1% of your magnesium is out for them to measure it in the serum. Hence, serum magnesium does not always accurately reflect total body magnesium stores. In fact, serum magnesium levels may be normal despite depletion of total body magnesium content. You may be low everywhere else, but your blood magnesium may still be okay. What do we say about vitamin A? Well, people will say my blood vitamin A is okay or it's low, but the research continually says that you could have way too much vitamin A in your liver even if your blood levels are low. So do you see the difference between a nutrient and a toxin? Vitamin A, they'll say you could be low in the blood and have tons in your liver. And here they're saying you could be normal in the blood and not have enough in the rest of your body. This is a vast difference between an actual essential mineral and a fat-soluble toxin. In experimental settings, total body magnesium stores can be estimated by measuring retention of an oral or intravenous magnesium load. However, measurement is cumbersome, so they're not going to do it and requires a 24-hour urine collection. Nobody's going to order that, except in research. In many instances, intracellular levels of magnesium, so they're talking about white blood cell measurements or red blood cells, serve as a better indicator for total body magnesium content compared with serum magnesium levels, with the most accurate test being blood mononuclear cell magnesium. Intracellular mononuclear magnesium content also correlates better with cardiac magnesium status. Funny thing that they don't mention hair magnesium levels at all. I can tell you, and I train people to tell you how well your topical magnesium or your pill magnesium is working based on a hair test where we compare it to hair calcium levels. It's not hard. Hair testing is the best indicator of magnesium levels. Red blood cell magnesium and even the white blood cell magnesiums that some places do are not good. I can have people whose red blood cell magnesium is high normal, and their hair test still says they suck, and they still have magnesium deficiency symptoms. Blood tests for magnesium sucks. Every blood has its advantages for certain things, and it sucks for certain things. Hair has its advantages for certain things, and it sucks for certain things. This is the nature of the beast. You don't go around trying to fix your entire house with only a screwdriver. Different tools for different jobs, and no one can seem to understand this. The hair test people want to say the hair test is the best for everything, and the blood test people want to say the blood tests are the best for everything. I don't understand why nobody can pull their head out of their ass and realize that certain things are good for certain things and certain things are better for other things. You don't try to use a screwdriver to do a hammer's job. These are simple concepts. And people would understand them in any other field, but in medicine, they just go dumb. Despite the importance of magnesium for the proper functioning of the cardiovascular system, surveys and studies have shown that dietary magnesium intake is often inadequate in the USA, which is consistent with the pattern observed in North European countries where osteoporosis is super high because they eat too much vitamin A. Anyway, several factors were thought to be contributory, including the loss of magnesium during food processing, low magnesium content of vegetarian diets, metabolic effects exerted by pregnancy. Pregnancy is just a super toxic state. Osteoporosis medication therapy. Did you know that depletes magnesium? Alcoholism, stress, as well as the differing magnesium content in water. Human dietary requirement for essential minerals such as magnesium is not precisely known, but I can show you how your magnesium is doing on a hair test very easily. You know what? Let me finish magnesium, Joe, and then I'll consider it. Okay, dietary magnesium and cardiovascular disease. So how much, if you're putting in more magnesium, how does your heart disease do? So this is how we look at things. We go, okay, if deficiency shows problems and either a higher intake or supplementing it fixes those problems, we can be pretty sure that we're on to something. Okay. If you take it away and you have problems and you put it back and the problems go away, you can be pretty sure that things are working. Dietary magnesium and cardiovascular disease, a review with an emphasis in epidemiological studies. You're not going to get metabolic ward, placebo-controlled, what's the one, crossover. You're not going to get that stuff. People are not going to live in a metabolic ward where everything that goes in their mouth and all their pee and all their sweat and all their poop and everything is recorded for like a year. You're not going to get that. People who throw out epidemiological studies or large-scale population studies, when they just throw them out on their face, they're missing out. We aimed to review current literature on circulating magnesium and cardiovascular disease, as well as potential biological processes underlying these observations. We concluded that a high magnesium intake is associated with lower risk of major cardiovascular risk factors, mainly metabolic syndrome, that's prediabetes, diabetes, and hypertension, stroke, and total cardiovascular disease. I believe that's CVD in this one. Higher levels of circulating mag, so higher serum magnesium, are associated with a lower risk of cardiovascular disease, mainly ischemic heart disease. That would be heart attacks and strokes, and coronary heart disease. So that is what we have there. How do we get, somebody asked, where do we get magnesium, or where do we used to? We didn't used to be as toxic. All these toxins deplete magnesium. Vitamin A depletes magnesium. I have a whole video on that here. Vitamin A depletes magnesium. Vitamin D depletes magnesium. Processing food depletes magnesium. The farmer's not putting magnesium back into the soil means you get lower magnesium in the plants they grow with that soil, and then in the animals that eat the foods that were grown in those soils. Like it's just a drastic thing. So for people who Let me close this down. Right now, we're ending this presentation. We're going to do a part 2 next week. I got to shorten these. So let me see what I have to go over next week, just so you can get excited. Okay, we're going to go over potassium in heart disease. We're going to go over zinc in heart disease. We're going to go over selenium in heart disease. We're going to go over niacin in heart disease. We're going to go over lactoferrin in heart disease. We're going to go over sodium versus potassium. And yeah, that's what we're going to go into next week. So next week will really be the nutrients. We did magnesium today. Next week will be the nutrients that we use, that we talk about to help with heart disease. So let me go back to Joe. I'm going to stop sharing and I'm going to get to this one. I'm not doing any more super chats. I'm doing the one super chat that's here, and then we're going to wrap up. Okay. So let me get back on. Don't send any more super chats. They're going to go to next week. Okay. So Just Man says, my opinion on meldonium/mildronate. Is it toxic? I had insulin resistance and it was helpful fixing that along with the Love Your Liver program. Thanks. Well, I got to do a little research. Because I don't do drugs. Sometimes I got to look these things up because I'm just not, that's not where my energy is put every day. Mildronate is a cardioprotective drug that improves cardiac function during ischemia and functions by lowering L-carnitine concentration in body tissues. That's an interesting mechanism. Let me see something. Well, the little bit of research that I'm looking at here, effect of long-term mildronate treatment on cardiac and liver functions in rats. They say long-term here, and they're saying they didn't see any liver dysfunction or heart dysfunction with long-term administration. Am I going to encourage taking pharma meds that somehow affect L-carnitine in a negative way? I'm going to say no, but if you took it and you're okay. Somebody asked me this the other day. They're like, well, what if somebody had taken, they're like, well, I have a friend who took ivermectin X amount of times. How do we know what amount of damage it caused? You don't get to know. The only time you get to know that a medication caused you significant damage is once the damage is present. So he said, how much is it going to cost to fix my car if it gets in an accident? Get in the accident and then we'll get an estimate. Like, how much poison does it take to poison someone? I don't know. The only way you find that out is to poison yourself. How many bullets can a person take before they're not around anymore? I don't know. Depends on the caliber, depends on where they get tagged, depends on all sorts of things. Depends on the help they get after they're punctured. So no, we don't get to predict these things. Could there be something long-term that happens from mildronate, like longer term? I don't know. I'm not going to try to scare you about it. What's done is done. If it helped you for a bit, okay. Am I going to advise other people to do that? No. So we're at basically, is it toxic? It doesn't look super toxic. It doesn't look obviously quite toxic. But fixing your vitamin A toxicity and copper toxicity, if you haven't watched the diabetes episode, I would watch that because those are the long-term things that are going to fix insulin resistance. Like vitamin A causes insulin resistance. It's all over the literature. So, oh gosh. David, can you put that link up? That meldonium link that you have? Or wait, maybe he can't. Here, let me search for that. Here, I'll just search for your quote there, David. I think it's just Wikipedia, right? Looks like it. Okay. Okay, I'm going to share this and then we're going to wrap up after this. Remember, no more super chats. Let me go share. I need that to move out of the way. Okay, there we go. Not this one, Joe. Okay. Now, for anybody who wants to have a fit that we're using a Wikipedia thing, look, I just need something quick. I'm live here. Give me a break. Okay. Oh, let me make this a little bigger. X-ray, crystallographic, and in vitro biochemical studies suggest that meldonium binds to the substrate pocket of gamma-butyrobutane hydroxylase and acts as an alternative substrate and therefore a competitive inhibitor. So what's the first word that we find here in terms of what do we talk about? Sometimes short-term improving people, but long-term causing them problems is things that inhibit pathways in the body. They inhibit detox pathways. So here we are. We already have the word inhibitor. Normally, this enzyme's action that it's a competitive inhibitor of on its substrates, gamma-butyrobutane and 2-oxoglutarate gives. So normally this enzyme, in the presence of the further substrate oxygen, the product's L-carnitine, so it makes L-carnitine, succinate, and carbon dioxide. So okay, I want to be sure here. Somebody tell me if you think I'm interpreting this wrong. In the presence of this alternate substrate, that being meldonium or mildronate, this reaction yields malonic acid semialdehyde, formaldehyde, dimethylamine. I believe, Will, that's an aldehyde also, right? And acetic, or this 1-methylmidazoline 4-il acetic acid. An unexpected product with an additional carbon-carbon bond resulting from N-demethylation coupled to oxidative rearrangement. Okay, anyway. Meldonium's inhibition of this enzyme. So, it's an enzyme that, when it's broken down, so they're not making, so these are things you're not making. You're not making L-carnitine, succinate, and carbon dioxide. Oh, the Ray Peters are going to be really upset that you're not making carbon dioxide. But then, when you put the meldonium in, or the mildronate in instead, it inhibits that enzyme, and the reaction now yields this aldehyde, I believe this is an aldehyde, and then whatever, the acetic acid that's bound to other things. Oh, sorry, the link. Let me get the link. It's just the meldonium for the wiki. Okay. Will says it's not an aldehyde. What was that, Will? What was the related compound that creatine breaks down into? One of those aldehydes that you mentioned. You remember, Will? Will did this thing. So creatine, especially when people took in huge amounts of creatine, which, funny thing, people on Twitter and everywhere else are starting to say, like, take 10 grams of creatine a day and there's no risk at taking high doses of creatine. And then Will posts, he can find it, but he doesn't have it right now, but he posts that there's several studies showing that when you take basically too much creatine for your body's ability to process, it turns into aldehydes. Now then people are going to go, should we not eat red meat? No, I didn't say that. It's when people are taking stupid high doses of creatine. They actually showed that when people took little, like, I think it was 2 gram doses or 1.5 gram doses or even 1 gram doses, they didn't make these things in their urine as much, nearly as much as when they took like a 10 gram or a 20 gram dose. So when you overload your detox systems, you get more toxic things at the end. Methylamine, that's what it was. Okay, yeah. The quote Will posted, creatine is a chemical that is metabolized to methylamine. Sorry, I got that confused with dimethylamine. Honest mistake, which is then converted into formaldehyde. Yes. So if you want a high dose creatine, yeah, your kidneys are probably not going to like it over time. Take the risk if you want. Am I worried about red meat creatine? No. If you decide you want to take creatine, I would suggest you limit it to 2 grams a day, no loading phase. I don't care what the swole bros on Twitter think of my recommendation. I don't. Too many of them fall apart and then you never hear of them again. So do I care what they think? No, I don't. Oh yeah, Will says dimethylamine is 2 methylamines, right? So can dimethylamine, let's go look that up. Wait, let's go see. Dimethylamine formaldehyde PubMed. Let's see if it breaks down to that. Well, let's just hit this real quick. This will be the last thing. So TMAO is something that they look for like, I believe it's heart disease. It's one of the markers that they'll look at for heart disease. So let's see, what was I going to do? I was going to look for dimethyl. Okay. TMAO is reduced to dimethylamine and formaldehyde. So trimethylamine N-oxide is broken down to dimethylamine and formaldehyde. Yeah. I don't know. I'm going to tell you, be careful with your creatine dosing. Be careful with it. Okay, so we're going to wrap it up for today. That's what I got for today. We're going to go over the nutrients of cardiovascular disease next time, which are all the same things. If you're in the Love Your Liver program and you're already following these recommendations, then you've got it done. Yeah, we'll go over that. It's the same stuff that we always go over because it is the same concepts and principles of toxic bile paradigm and toxicities and deficiencies that are the cause of all chronic disease. So if you guys go and you watch the other live streams where I go over different diseases, and I will continue doing this, you're going to see the same patterns of the same toxins, the same bile problems, the same microbiome problems, and the same nutrient deficiencies over and over and over again. People will be like, well, you can find anything in literature. How could I find these same patterns over and over and over again in all these chronic diseases? Seems kind of weird, right? So, oh yeah, Pierre asked, why isn't it called methyldehyde? I don't know. To hide it? I don't know. So, okay, so let me hit the links. I think Joe's been showing them like rapid action. Okay, so if you want to find our main website, nutritiondetective.com, that's where you can find if you want to do a consultation with any of our facilitators, me included. That's where you can find that. Always remember that if you get a package with one of our facilitators, our health facilitators, you may do the consultation with them, but you can always come to my office hours and get support and troubleshooting from me directly. You do have access to me through the office hours, which I do once a week, and mine's usually pretty busy. So anyway, so there's that. Then we also have our supplements there. We are just trying to get out the new batch, and then we're going to start on a whole new batch of stuff. We got 5 products that we're trying to get out. It's just, the delays. That includes, don't email Julie. That includes the liquid zinc picolinate. Don't email Julie about it. I'm going to have her reply to you and say, Dr. Smith said, don't email me about it, but we are working hard on it. Okay. And Kelsey's zinc niacinate salve, and it's awesome. I started using it regularly myself because I'm trying it in a new way. Pretty excited about it. So anyway, those are coming out soon, and then we'll be getting more of Kelsey's products out after that, and other stuff to try to make it easier for people to dose themselves in smaller doses or in higher doses or any of that stuff that they want. And then if you want to find the Love Your Liver program, that is members.nutritiondetective.com. We sent out a newsletter the other day about what's happening right now is the price of, okay, so right now it's $99 a year. As of today, January 28th, 2025. So do not email Julie later if you miss this opportunity, because we've been sending it out on email, newsletters, and everywhere. Right now it's $99 a year. Okay. Yes, it is a subscription thing. We are changing that. We are changing it so if you're already in it, if you are in it when I switch it, when I flip the switch, which is coming, when I flip the switch, it's going to $199 lifetime. So there's not going to be a subscription anymore. It's going to be $199 lifetime. It's doubling in price, but you get it for good. So if you're already in it when I flip the switch, and you're grandfathered for life. Okay. If I flip the switch, we've already extended this like 3 or 4 days now because people are rushing to get in. So we've already extended it a lot. So if you miss this, I'm sorry. Don't email Julie. You've had plenty of time to join the program. You've probably had months or years to join the program and you never pulled the trigger, and now I'm changing it. But I'm changing it for life. So then you only have to pay one time instead of yearly. Okay. And those of you who let your subscriptions lapse, you want a lifetime membership, you're probably going to have to rejoin. People who do the consultation packages with us get automatic lifetime membership with the package purchase. That's always been the case. So, oh yeah, Hound Jog, we send out 2 or 3 emails about it this weekend. So if you're not getting them, I'd suggest you check your spam. Yeah, because we send them, or maybe you're not on the newsletter list. Yeah, that's at the bottom of the last 2 or 3 newsletters. William's been making those, and I've been reviewing them, and we've been sending them out. So they are going out. Yeah, as Soil Food Web Warrior says, lowering the price is insane. Yeah, let me tell you, folks, I seriously, I didn't want to do a yearly subscription to the Love Your Liver program. And I listened to a business advisor type who said you should do this. And I did it. And I didn't like it because I personally do not like subscription stuff. I will purposely not join things that have a subscription type thing. I didn't want to do it. They convinced me to do it. And Julie and I talked about it. And we were like, we're not working with that guy anymore. And we were like, let's kill it. Let's get rid of it. I don't want it. This is not the way I do things. So yeah. So it's going to be $199 lifetime after this. If you're already in it, if you're in the subscription model before I flip the switch, then you are grandfathered into lifetime stuff. So that's happening. So if you miss it, you miss it. I've already extended it a bunch, and you have a chance today to do it. I'm probably flipping the switch tomorrow, so I have all day to make sure that all of this is working. Okay. So, yeah, so I got to go do my inner circle now. That's inside the Love Your Liver program. That's private Q&A where it's all Q&A every time. And the people who are in there get to ask the questions. So I will see you all next week. Have a great week. Oh, there's Beth Martins. Hi, Beth. And thanks for getting me on the Missing Link podcast yesterday, Beth. That was a lot of fun. That's like a 2.5 hour, 2 hour, 45-minute podcast. So you guys may want to watch that. We did get into tanning stuff a lot of light stuff. So anyway, have a great day. Have a great week. And I'll see you next week. I don't know what the topic is. Oh no, the topic is obviously it's part 2 of this. So duh, I will see you all then. Bye now.